Hello, I am Carlo Colantuoni, and I'd like to tell you today a little bit about NeMO Analytics, which is a web portal where we have collected as much public data in transcriptomics and epigenetics focused on the development of the neocortex as we could, and making it available to both computational biologists for open analysis and for biologists without coding experience. To explore at the gene level, and as I'll show you, gene signatures as well. So in other talks, I've covered this paper in more depth. It's a paper that's part of an upcoming BICAN package for the developing brain in the Nature family of journals. That's the QR code at the lower left. The methods we use to break apart all this data are detailed in the paper at the lower right. I won't talk too much about the methods today, but I have other talks on those in that other paper. In other talks, I've covered the sort of overarching structure of the paper, which really starts from development looking across species for conserved elements of neurogenesis. But about halfway through the paper, we shift and actually begin the investigation from adult data and then look back in development to see when particular neuronal identities emerge. And what I want to cover specifically today is the epigenetic control of some primate-specific signatures in the cortex that we first observe using these diverse data resources and then investigate more deeply looking at some epigenetic data. And all of this is really an example of how much we can see and how many questions we can ask by putting two really important things together. One is the data collection. We collected as much public data, transcriptomic and epigenetic data across the development of mouse, monkey, and human neocortex, as well as in vitro stem cell systems modeling this process in a dish. And the second thing is gene signatures of interest, whether that's a packet of genes that you know are controlled by a transcription factor that drives a particular neuronal laminar identity, or as I'll show you, things we decompose when we jointly harness many of these data sets to see common signals. Once you put those things together, a giant data collection and gene signatures of interest, you can really ask a lot of questions and get a lot of answers quickly. And that's really what NeMO Analytics is designed to do. And I'll show you all the figures I show today are screen captures from NeMO Analytics. So you could do all of this except the fancy decompositions I show by point and click. And the fancy decompositions are in the methods paper if you want to get into that. But of course, you can capture your own decompositions or gene lists and bring those to bear on this. So hopefully all of that will make sense by the end of this talk. Let's go. Okay. So as I mentioned, the beginning of the paper starts in development, but where I want to talk about today in these laminar identities begins from adult human data. So we'll get into what all of those panels on the right are, but first we grab two very large human adult studies from the Allen Institute, single nucleus RNA sequencing. We only used the SMART-seq data sets, so that's much deeper sequencing than the more common 10x or droplet-based methods. Costs a lot more, but you get a lot more deep sequencing and actually see what cells are doing rather than only being able to classify what they are. We want to know what they're doing too. Okay, so we only took glutamatergic neurons, the deep sequencing data. The two studies span 5 different donors and 8 regions. And we used our structured joint decomposition package in the R programming language to break apart these data. And it's the joint NMF function that we used, which takes those 5 donors, each as a single matrix of RNA-seq data, and asks the question, what are some common sources of variation across those 5 matrices? Now, because we only included glutamatergic neurons across regions and across layers, inside each donor's matrix are all of that variation. Different kinds of glutamatergic neurons in different places doing different things. So the signals we pull out will be variation that is observable in all 5 of these donors across regions and layers. So we asked the joint NMF to give us 5 signatures or dimensions that together can be used in different combinations to explain all of the gene expression patterns for every single gene that are measured in these studies. Okay, so now we're getting to what the picture at the right is. The 9 rows are 9 of those 20 transcriptomic signatures, and the 7 columns, well, there's actually 8 columns, but there's 7 data sets. Each column is a data set where we're looking at each one of those laminar identities that we extracted from the first 2 data sets. So that's a projection of the signatures we get out of the first 2 data sets that have all the deep sequencing, all adult neurons. And we're going to look at that across different mouse, monkey, and human data sets, and spatial and single nucleus data sets. So I'm going to highlight 2 of these. They're arranged in order of depth in the cortex. So we have layer 6 signatures and to 5, 4, all the way up to layer 2 here. And you can see them in the cortical slices here. So what I want to do though is zoom in on 2 of these in particular. But I wanted to show you all of them before we leave, just to show that really the data from the ALAN combined with these joint decomposition techniques was really able to give us a rich spectrum of transcriptomic signatures describing the diversity of neurons in the neocortex across the adult human. So a few notes that I'll cover in more depth in other talks before I move on to the next slide. These signatures take many years and even into the first decade of life to reach their maximum levels in the human cortex. So in other developmental data sets, we take these adult signatures and look at them emerge, and it takes them a long time to come up. And that is in stark contrast to some of the master TFs, the master transcription factors that drive laminar identities in development, which are high very early in fetal life and actually come down over fetal development as these maturity signatures that we're looking at come up. So it really looks like the TFs like RORB, that is a layer 4 signature, FESF2, which is a deep layer signature. They are able to initiate the structure of these laminar specific maturity signatures in neurons and set up an epigenetic environment where they themselves, high levels of their own RNAs are not needed throughout life to maintain those. They actually go away while the mature laminar specific signatures are maintained throughout life. We also go in to show that in organoids, again, the transcription factors that initiate things are often expressed, but the maturity signatures don't come on. That can be somewhat remedied by xenotransplantation into the brain, by putting organoids in the brain of a young mouse, for instance, but not entirely. So that's some just teasers that are things in the elements in other talks in the paper. But we're going to look at layer 4 today. Okay, so this is just those 2 rows of data that we were on the other slide, but zoomed in. So in this Visium data, you can see that the layer 4 signature is kind of a pinstripe. In the Merfish data too, a very, very exact layer of cells in layer 4. The layer 3-4 signature is a little more diffuse, rising up off of layer 4 to have kind of a range there. Those are in 2 different spatial data sets from the human, where we have low spatial resolution, high transcriptome resolution in Visium, and Merfish is a high spatial resolution, but only, say, a couple hundred genes. And then the third one here is now in the adult macaque, so a whole hemisection of the macaque brain, where they're using stereoseq here, which gives you a little bit of the best of both worlds, where you have high transcriptome resolution and high spatial resolution. And you can really see that the signatures we pulled out of single nucleus data really localized to extremely specific neuronal populations in these specific layers. The next column starts to get interesting. We're now in the mouse. We can see this layer 3-4 signature very clearly there. It appears it's also shared with a neuronal population in the thalamus, but the layer 4 signature in this row is absent entirely from the mouse cortex. And we can see that is validated if we look at more data where we have human, chimp, and mouse data from different studies here. The conserved signature here shows up in the mouse, it shows up in the chimp, and it also shows up here. Actually, this is human, chimp, and mouse. And if we look at the layer 4 signature down here, human, chimp, and the mouse does not show it. So we're quite confident that this is a signature that is in the chimp and human layer 4. It is not in layer 4 neurons of the mouse. These two studies, this one here and this one, actually noted in those publications a layer 4 primate-specific signature. And we looked at the genes they had identified in that signature, and we have here identified the same one. They did not realize that they had mutually discovered that same thing, but it's very reproducible, and it's interesting in that it is primate specific. And so we wanted to go look a little deeper into it, and it's epigenetic control. And to do that, we used yet another incredibly cool data set that is in the public domain, again, to show the ability to ask questions and find answers once we bring really diverse data sets together. So there's a lot of panels here. Let's take it slow. This is from Rong Fan's lab at Yale. Di Zhang is the first author. This is in adult mouse brain here. And each two pictures that are in a dotted line together are multi-omic data from the same slice. For instance, this was an RNA and ATAC experiment, so you're getting multimodal transcriptomics and epigenetics from the same slice. This was the RNA data from that experiment, and this is the ATAC data on that same section that RNA and ATAC were generated on. Here we have RNA and H3K4 trimethyl that were done together. And again, you got to watch those dotted lines to see which are the slices that were studied together. And here we have H3K27 acetylation along with RNA. And then the one we'll also end up being interested in is at the bottom here, they're separated, but they're still within one of these dotted lines. So here we have the RNA plus H3K27 trimethyl. And here's the RNA for it, and here's the H3K27 trimethyl. Okay, so to bring this cool multimodal spatial data to bear on the signatures we just looked at, we projected both the layer 4 conserved signature, which was the layer 3-4 signature, I believe, and then the layer 4 primate-specific signature into these data. So that means we are going to visualize the RNA or ATAC or H3K9 data at the transcriptional start sites for the genes that came from our transcriptional signature. So in the conserved signature, as we would expect, you can see the RNA and you can see it in the open chromatin at those same TSSs. So TSSs, transcriptional start sites that were associated with our layer 4 conserved signature are open, and they're expressing RNA in these slices, as we would expect if everything were working as we would expect it to. The same is true for the RNA and H3K9 trimethyl, where we have RNA here and we have the H3K9 trimethyl here. So both of these are, either open chromatin or the histone mark associated with gene expression. Okay, then let's look now at the RNA and H3K27 trimethyl. Again, for the same signature, the conserved one. The RNA is there again, as we expect, in that layer 4. But now, in the H3K27, the repressive histone mark, where that layer 4 signature usually is devoid of signal up here. Is that in the lower layers of the cortex, this conserved layer 4 signature is actually repressed via the H3K27 trimethyl signature. So it's up in layer 4, it's down in the deep layers, the histone controlling marks. Now, this is where it gets interesting, is when we start to look. So this is all what we would expect if this is a conserved transcriptomic signature, mouse monkey in here, in the mouse, and the mouse monkey in human. So the positive marks follow the RNA and the open chromatin. It gets interesting when we look at the layer 4 signature that is not in the mouse, that is primate specific. So all of these panels on the right are the same as they were on the left, but now looking at our primate specific instead of our conserved signature. So in the RNA-seq, in all of these experiments, no matter what they were coupled with, there's no specific signature in the cortex, as we would expect. That's how we got here. We saw it was not in the cortex of the mouse. Similarly, in ATAC, there's no specific layer signature. H3K4 trimethyl, no specific signature. But now the H3K27 trimethyl signature is quite interesting. So this is the RNA from that experiment, no signal. Here is the H3K27 from that coupled RNA sequencing and H3K27 signature. Here's where it gets interesting. There is a specific signature in the mouse to repress this program in the layer 4, and it appears really some of the other upper layer identities in the mouse. So what this means is that the transcriptomic signature is not simply absent from the mouse. It is not something that did not evolve until primates were around. So likely it could be used somewhere else. But it is one that is not used by the mouse in that layer. This is in some ways in keeping with what we know about evolution, that it is very rare that a mouse has a cell type, a liver cell type, a kidney cell type, any cell type that is not in other mammals. We look a lot like each other in different shapes. So in this case, it appears it is not a transcriptomic program that was invented in the primate lineage, but rather a transcriptomic program that was invented earlier in evolution, but simply used differently in different places. And in this case, it is being used specifically in the primate in layer 4 neurons and not there in the mouse, but actively not there, actually shut down specifically to not show up in layer 4 neurons. What this is doing and why, we'd have to dig deeper. That will come back to the genes underlying these, this layer 4 signature, the primate specific one, actually includes a gene called FOXP2, which is involved in language development in humans. So it is quite interesting that even though it may be an old evolutionary transcriptomic program, it is a new use of that program in neurons that it's not being used in the mouse, but is now being used in primates, eventually in neurons that in the human are fundamental in the learning of language. So again, to take a step back, all of this is basically to show how we can use large data collections that are very diverse in what they're studying, whether it's in a dish or different species, different regions of the brain, and either decomposition techniques to get gene lists of interest or existing lists of genes that we all have From a lab experiment, what are the downstream genes of FezF2? Where and when do they come on in different species? Once you get the data sets together and gene signatures together, you're off to the races with questions you can ask.